Today, we'll be talking about induced pluripotent stem cells and their applications for disease modeling and cell therapy for epilepsy. But first, what are IPSCs? IPSCs are a stem cell derived from adult tissue. Typically, skin cells are taken from a patient, and these cells are reverted back to a state resembling early embryonic development. From this state, these stem cells can be differentiated into any cell type found in the adult body. Here, we see IPSCs in culture turning into neurons. This allows researchers to grow human brain cells in a dish in order to study their structure, function, and development. So how can iPSC technology be used to study epilepsy?
Previously, embryonic stem cells were used because they can turn into any cell type in the body. However, there are ethical issues that arise due to their source. Now we can get stem cells from adults by taking skin or blood cells and reprogramming them. This is a much less controversial method. One of the common ways iPSCs are used is to get skin or blood cells from a patient with a known disease and then reprogram them back into iPSCs. Then they can be differentiated into disease-specific cell types to study the disease in a dish. These cells have the disease-causing genetic background, and you can get controls from siblings that do not have the disease. Although this is very useful, they can be difficult to obtain for rare diseases. However, they can be used to study diseases without a known genetic cause, such as psychiatric disorders like schizophrenia. Another way that iPSCs can be used is to take skin or blood cells from someone without any known diseases and then edit them using gene editing technologies such as CRISPR. This can be used to knock out a gene, to repress a gene, or activate one, or to insert a specific mutation. Once we have the edited iPSCs, we can turn them again into the disease-specific cell types to study the disease in a dish. As the skin or blood cells can be taken from a patient without any known disease, they can be taken from almost anybody. And although the cells may not have the genetic background that caused the disease, they are good for studying diseases that have specific genetic causes. Epilepsy, for example, has a strong genetic component. This is an example of how we can combine these two methods to study epilepsy. First, we can get skin or blood cells from an epilepsy patient and turn them into iPSCs. And then we can turn those iPSCs with specific epilepsy mutations into neurons that also have the epilepsy mutation to study them in a dish. Additionally, we can use gene editing techniques like CRISPR to correct the iPSCs and create a control that is almost identical to the diseased neurons. After we have the neurons in a dish, we can study how they behave. This is an example of how neurons communicate via action potentials. 70% of epilepsy patients respond well to medication. However, there are some patients who do not respond to treatment. We can use iPSCs to screen novel drugs and find better treatments for patients who do not respond well.
Another application of iPSC technology is to generate cells for transplantation therapy. The loss of certain cell types in the brain can cause epilepsy, and previous work in animals has shown that replacing these cell types can stop seizures and restore learning and memory deficits. In this next video, we will see the behavioral effects of these stem cell transplants. A typical mouse will not show aggression or hyperactivity in response to handling, whereas an epileptic mouse does. Here, we see an epileptic mouse being handled and attacking the handler. Epileptic mice receiving stem cells 60 days after transplantation show a 90% reduction in seizure frequency and a normal handling response. In order to produce the cells needed to treat humans, we must use iPSCs. Fibroblasts, or skin cells, can be programmed into iPSCs. iPSCs can then be used to generate any of the cell types present in the adult brain. Researchers around the world are currently working on protocols in order to generate each of these specific cell types. IPSCs allow researchers to study epilepsy in ways never before possible. By growing human brain cells in a dish, we can study the structure and function of epileptic neurons derived from patients. We can use gene editing to investigate the role of specific mutations. And we can also use IPSCs to generate cells for therapy.